Good day, everyone. I'm Vernice Gallano, together with Marlie and Tullao, and we will be presenting a research entitled In Silico Evaluation on the Binding Affinities of Steroidal Bufadienolides from Toad Venom on Fc gamma and death receptors. According to the Global Cancer Observatory, there were approximately 18.1 million reported cancer cases worldwide in 2020. This alarming number is expected to rise in succeeding years. In fact, the World Health Organization predicted that global cancer cases would reach to over 30 million by 2050. This increase in cancer incidence highlights the urgent need for an effective and accessible treatment options. While conventional treatments such as chemotherapy and radiation remain the standard of care, these approaches are often associated with severe side effects and limited selectivity. In recent years, researchers have begun exploring natural compounds as potential sources of new anti-cancer agents. Among these, animal venoms, specifically those derived from toads called bufadienolides, which are steroid-like toxins, have shown promise. These venoms are rich in bioactive compounds known to induce apoptosis, or programmed cell death, which is a key mechanism for eliminating cancer cells.
In most studies for cancer treatment, their main focus are on death receptors due to their role in apoptosis, causing Fc gamma receptors to be overlooked. Similarly, papers that delve deeper into the probability of bufadienolides as medicine mostly use bufalin and arenobufagin, disregarding other bufadienolides derived from toad venom. Furthermore, there is a lack of comprehensive in silico screening for toad venom-derived bufadienolides. There is also only a few number of studies that evaluates combined receptor targeting. In terms of cost and efficiency, in vitro and in vivo studies demands more time, effort, and money compared to in silico evaluation.
The objective of our study is to determine the dual targeting capability of toad venom-derived bufadienolides towards Fc gamma and death receptors for potential cancer treatment through in silico methods including toxicity screening, ADMET profiling, molecular docking, molecular dynamics, and thermodynamic analysis.
Huachanse, derived from toad venom from Bufagardizans, is a drug approved in China for cancer treatment since 1991. Aside from this, it is also utilized as an anesthetic and cardiotonic agent. In 2005, it was clinically tested in the United States, which showed that it stabilized lung and liver cancer tumors by 40%. A study of Liang et al. In 2022 tested the effectiveness of bufadienolides, specifically sinobufagin and bufalin, against hepatocellular carcinoma, also known as liver cancer. They discovered that the mixed group has a tumor inhibition rate of 88.49% through in vitro testing. The results also demonstrated an increased apoptosis, indication of necrosis, and dissipation of tumor nucleus and mice tumor cells. A clinical trial conducted by Mussolino et al. In 2022 found that Fc receptors should be the basis for monoclonal antibody therapy due to positive results for anti-tumor reactions in breast cancer. Additionally, a study by Lee and Ravech in 2012 showed that death receptor 5 antibodies require Fc gamma receptors to further promote apoptosis through in vitro and in vivo trials.
Starting on the left side of the workflow with the bufadienolide compounds, which we sourced from different online databases. These compounds were then loaded into Protox2 for initial toxicity screening. We selected only those with toxicity classes between 4 and 6, which are considered moderately toxic but still viable for drug development. Further ADMET evaluation was performed using ADMET Lab 3.0 to assess drug likeness and pharmacokinetic properties. Simultaneously on the right side, we focused on our targets, death receptors and Fc gamma receptors, which we retrieved from protein data bank. These structures were cleaned and prepared by removing water molecules, followed by visualization in PyMol and UCSF Chimera to ensure structural accuracy. Next, we perform molecular docking using CBDoc2, which helped us visualize potential binding pockets and get preliminary understanding of how the bufadienolides interact with protein targets. We then docked ligands and receptors again using Autodock Vina for more precise docking results. From this, we selected the top 3 ligand receptor complexes based on binding affinity. For the top hits, we proceeded the molecular dynamics simulation using Romax 2023.2 for 10 nanoseconds. Prior to simulation, the systems were solvated, neutralized and assigned with the force field parameter using charm 36 to ensure compatibility with Chromax. We generated input files via charm GUI solution builder. Post-simulation, we analyzed the binding stability and energy using GMMPDSA, which provided insight into the thermodynamic favorability of the interactions.
Out of the 51 bufadienolides that underwent a preliminary toxicity assessment, only 7 that have a toxicity class from 4 to 6 are considered. This is implemented to exclude compounds that are considered lethal, while retaining those with pharmacological potential and are within the acceptable safety margin. Many of the existing and approved targeted cancer drugs remain viable despite their toxicity. An example of this is cetuximab that has an LD50 value of 300 milligrams per kilogram. A development of anti-cancer drugs with reduced toxicity and high selectivity is necessary as most of the cancer therapies available at present suffers from poor selectivity between healthy and malignant cells, which in turn affects various healthy organs and immune functions.
Our ADMET evaluation provided a comprehensive look into the pharmacokinetic and drug-like properties of the 7 bufadienolide compounds and served as our second screening step. Based on physiochemical criteria, 6 out of 7 compounds met major drug-likeness filters, including Lipinski, Gauss, Weber, Egan, and Moeg rules. The only exception was cinobufadienolide, which violated 3 of these rules due to its high molecular weight and structural complexity, making it less ideal as a drug candidate.
The binding affinity of the bufadienolides ranges from negative 4.6 to negative 12 kilocalories per mole. Most of them are equal to or greater than negative 7 kilocalories per mole, which is the acceptable range for good binding. It can be observed that resibufaginol consistently scored the highest binding affinity, while bufotenin performed the weakest.
So the results show that resibufagenin, which is the black line, demonstrated the most favorable profile, maintaining RMSD values similar to our positive control, showing minimal fluctuations in RMSF and exhibiting the lowest SASA values. This suggests stable, compact binding to the fast receptor. Marinobufagenin, which is the green line, showed intermediate stability, following trajectories similar to positive control in both RMSD and radius of gyration, indicating structurally favorable binding. Resibufaginol, which is the red line, displayed the highest variability across all parameters. Elevated RMSD fluctuations, the highest RMSF peak at 1.4 nanometers, maximum radius of variation at 2.38 nanometers around 8,500 picoseconds, and the highest SASA value. This indicates a more flexible, extended, and less stable binding conformation. Notably, cetuximab showed higher RMSD variations, confirming weaker binding compared to our bufadienolides. These results show that resibufagenin and marinobufagenin showed the most promising stability profiles for fast receptor binding, while resibufaginol appears less suitable due to its higher flexibility and instability. So, in the figure, we can see that resibufaginol and resibufagenin both closely tracked the IgG-Fc trajectory in RMSD, indicating stable binding with minimal structural changes. These compounds show promising therapeutic potential for Fc gamma receptor targeting. 11-hydroxyresibufagenin showed concerning behavior. Its RMSD continued increasing even at 10 nanoseconds, suggesting it failed to reach equilibrium. This indicates potentially unfavorable or weak receptor binding. The RMSF data supports these findings. 11-hydroxyresibufagenin exhibited the highest flexibility peaks, while resibufagenin showed the most constrained fluctuations similar to our positive control. Notably, the radius of gyration shows resibufaginol adopting a more extended conformation early in the simulation, while 11-hydroxyresibufagenin becomes more compact towards the end. The SASA profiles demonstrate good overlap among the stable compounds, indicating consistent solvent exposure patterns. Overall, resibufaginol and resibufagenin demonstrate stability profiles comparable to our positive control. The MC gamma receptor 3 simulations show remarkably consistent behavior across all bufadienolides. All three bufadienolides, resibufaginol, marinobufagenin, and 11-hydroxyresibufagenin, demonstrated excellent stability, staying well below zero point 0.4 nanometers RMSD throughout the simulation. Notably, all bufadienolides performed better than cetuximab, which showed a brief fluctuation peak at 3 nanoseconds. The RMSF analysis reveals that all bufadienolides are actually more rigid than the IgG1-Fc control, with peak fluctuations occurring at residue 170 rather than extending beyond residue 600 like the control. Radius of gyration data shows the bufadienolides maintain compact conformations compared to the more extended IgG1-Fc, indicating stable and tight binding. The SASA profiles demonstrate that all bufadienolides follow similar trajectories, showing consistent solvent exposure patterns that reflect their smaller size and stable binding. So for Fc gamma receptor 3, all three bufadienolides show excellent stability profiles, suggesting this receptor may be particularly amenable to bufadienolide binding. In summary, our dynamics results show clear ligand receptor preferences. Resibufagenin works well with all three receptors. Marinobufagenin is best for a fast receptor, and resibufaginol excels with Fc gamma receptor 1. Notably, Fc gamma receptor 3 accommodates all compounds well, making it an attractive therapeutic target.
It can be observed in the thermodynamic analysis of the bufadienolides that all of the values generated for the van der Waals energy are within the limits of the positive and antibody control, indicating good attractive forces between the molecules of the bufadienolides and the three receptors. For the electrostatic energy, only resibufaginol and marinobufagenin docked with Fc gamma receptor 3 are within the limits of the positive and antibody control. Despite this, all of the generated values are negative, which shows attractive forces acting upon the complexes. For the polar solvation energy, only 11-hydroxyresibufagenin docked with Fc gamma receptor 1 is within the limits of the positive and antibody controls, indicating that it has a strong stability. In the solvent accessible surface area, only the bufadienolides docked to fast receptor generated values within the limits of the controls, showing that they are more favorable, as this indicates that they were not highly affected by the solvent. For the total binding energy, only 11-hydroxyresibufagenin docked with Fc gamma receptor 1 is outside the limits of the controls, while the rest of the generated values are within the limits, emphasizing their stability. Nevertheless, all of the values are negative, indicating that binding occurred between the bufadienolides and the DET and Fc gamma receptors. For the entropic factors, all of the bufadienolide and receptor complexes generated a positive value, which indicates that it contributes favorably to the binding energy. On the other hand, 11-hydroxyresibufagenin docked with Fc gamma receptor 1 is the only bufadienolide that generated a positive value in the enthalpic factors. This indicates that it is endothermic and has weaker binding interactions. This may also be the reason why it is not within the limits for the total binding energy. In conclusion, based on the computational analysis conducted to the bufadienolides, they were detected to bind effectively to the DET and immune receptors, especially to the fast receptor, Fc gamma receptor 1, and Fc gamma receptor 3. Most of the produced values surpassed the antibody control, showing a strong binding between the molecules. Based on the structure-activity studies of specific bufadienolides, it was revealed that their activity as immunosuppressant, antineoplastic, and oxidoreductase inhibitor is highly probable. Additionally, the ADMET evaluation of the bufadienolides revealed that they exhibit drug-likeness and can be absorbed through the gastrointestinal tract. It is recommended that future studies are in vitro and in vivo for experimental validation. Subsequent studies that aim to expand the ligand library are also recommended, as there is a lack of public chemical databases of bufadienolides. Additionally, in future in silico studies, it is suggested to include a known smaller molecule inhibitor that binds to the same receptor for a more suitable control sample. In the simulations, it is advised to extend the molecular dynamics simulation from 10 ns to 100 ns and incorporate molecular mechanics generalized Born surface area alongside molecular mechanics Poisson-Boltzmann surface area to have a better capture and better perspective on the interactions of the complexes. Lastly, the incorporation of a well-defined antibody controls with no expected interaction in future studies is recommended to serve as a baseline for non-specific binding.